The overall goal of this procedure is to quantify the binding of short peptides with human MHC II immune proteins using a high-throughput 384-well plate assay. This is accomplished by first performing solution phase competition binding assays with the peptides of interest. In the second step, the equilibrated peptide MHC II complexes are captured with an anti-MHC II antibody immobilized on high-binding ELISA plates. Next, the captured complexes are incubated with europium-labeled streptavidin, and then the unbound streptavidin is washed away and the captured europium is activated. Ultimately, time-resolved fluorometry can be used to quantify the levels of MHC II captured control peptide.
This method can provide preliminary insights into the immunogenic potential of target proteins. It is particularly useful for validating the output of in silico epitope predictors.
Begin the procedure by adding 25 microliters of freshly diluted L243 antibody solution to each well of a 384-well high-binding white ELISA plate. Seal the plate with a polyester film and incubate it overnight at 4 degrees Celsius. Then, starting with a 10 millimolar stock of each test peptide in DMSO, make the following dilution series in citrate phosphate buffer using 384-well polypropylene plates.